Welcome to Microscopic Marvels.
All right, let's jump right in. Today, we're going to tackle one of the most fundamental, most powerful tools in all of microbiology, the gram stain. This single technique has been a cornerstone for over 100 years, and trust me, understanding it is absolutely non-negotiable, not just for your exams, but for real-world medicine.
So I want you to imagine this. A patient comes in, they're really sick with a bad infection. How does a doctor get that first crucial clue to pick the right antibiotic? We're talking hours, sometimes even days, before the final lab results are ready. Well, the answer, so often, starts right here with the test we're about to break down.
So, here's our game plan. First, we'll cover the basics. What exactly is gram staining? Then, we'll get into the science of why it actually works. After that, we'll walk through the procedure step by step, figure out how to read the results, and finally, tie it all together to see why this old-school test is still a giant in modern medicine.
All right, let's get into it. Section 1. What exactly is this test, and why is it so important?
The gram stain is, without a doubt, the most widely used stain in bacteriology, period. It was developed way back in 1884 by a physician named Hans Christian Gram. And its job is beautifully simple. It helps us see bacteria, and more importantly, it helps us classify them into two huge distinct groups.
So you'll hear it called a differential stain. Why is that? Well, it's because the whole process uses different dyes to make different types of bacteria show up as different colors. And that color difference is the key. It lets us sort almost all the bacteria that matter into two big families, the gram-positives and the gram-negatives.
Okay, so that leads to the million-dollar question. Why? Why do some bacteria hold on to one color while others end up with a completely different one? The entire secret, it turns out, is hidden in their cell walls. It all comes down to the cell wall. On one hand, you have gram-positive bacteria. Think of their cell wall as a big, thick brick wall. It's made of a substance called peptidoglycan, and we're talking 50 to 100 layers thick. But then you have the gram-negative bacteria. Their peptidoglycan wall is super thin, like only 1 or 2 layers, and it's covered by an extra outer membrane. This single difference in architecture is the key to the whole shebang. So let's see how this plays out. First, we hit all the bacteria with the primary stain, crystal violet. At this point, everybody turns a deep violet color. Next, we add iodine. The iodine acts like a glue or a mordant, forming a big complex with the dye inside the cells. Now, for the game-changing step, the decolorizer. When alcohol hits the gram-positive bacteria, their thick wall dehydrates and shrinks, trapping that dye complex inside. They stay violet. But the gram-negatives? That alcohol just dissolves their outer membrane, and the thin wall can't hold on. Poof! The dye washes right out, leaving them completely colorless.
Okay, so now that you get the why, let's walk through the how. This is the actual step-by-step procedure you do in the lab.
And for all you exam takers out there, here's a super high-yield mnemonic you gotta remember for the order of the reagents. It's CIAS. That's crystal violet, iodine, acetone alcohol, and safranin. CIAS. You gotta lock that one in.
Getting these 4 steps right is everything. Step 1, you flood the slide with crystal violet for about a minute. Everything turns violet. Step 2, you add grams iodine, the mordant, for another minute to lock in that color. Step 3, here comes the all-important decolorizer, but only for a few seconds. And finally, step 4, you add safranin, the counterstain. This is what gives those now colorless gram-negative bacteria their nice pink or red color.
Now listen up, because this is a classic exam question. I cannot stress this enough. Decolorization is the single most critical step of this entire process. If you decolorize for too long, you'll strip the violet dye from everything, making it all look pink. That's a false negative result. But if you don't do it long enough, everything stays violet, giving you a false positive. Timing here is absolutely everything. Okay, so you've done the stain perfectly. You look through the microscope. What do all these colors actually mean? Let's figure out how to interpret the results.
Here's how it breaks down. If you're seeing deep violet or purple bacteria, you're looking at gram-positives. They resisted that decolorizer. Think of your classic cocci like staph and strep, or rods like bacillus. On the other hand, if you see pink or red bacteria, those are your gram-negatives. They got decolorized and then soaked up that pink safranin. Classic examples here are things like E. coli and salmonella.
All right, time for some important exam alerts. Because not every bug plays by the rules. You've got organisms like mycoplasma. They don't even have a cell wall, so the stain has nothing to stick to. Then you've got the really skinny ones, the spirochetes, whose walls are just too thin to see with this method.
And don't forget the intracellular bugs like rickettsia and chlamydia that are literally hiding inside our own cells. And one more super high-yield fact for you. Yeasts like Candida will actually show up looking gram-positive, big violet ovals. Don't let that fool you.
So we've covered the what, the why, and the how. But let's bring it all home and talk about the most important part. Why does any of this actually matter when you're taking care of a patient?
The clinical power here is just immense. It's the very first step in identifying a mystery bug, and because many antibiotics work on one group but not the other, it helps the doctor make a smart, educated guess on the right antibiotic to start immediately. We call that empirical treatment. It's also a great way to check if a sample is even good enough for culture, and hey, it can even give you a quick heads up about a yeast infection.
Let's just follow the journey. A sputum sample from a patient with pneumonia hits the lab. We can do a gram stain in just a few minutes. The report comes back, gram-negative bacilli seen. Just based on that one piece of information, the doctor can start an antibiotic that's known to work against those types of bugs. This decision happens in minutes, giving the patient a massive head start on treatment while everyone waits for the final culture, which could take a day or two.
So I'll just leave you with this thought. In our modern age of DNA sequencing and all kinds of advanced molecular tests, why does a technique from the 1880s remain one of the most important tests in all of medicine? Well, the answer is simple. It's fast, it's cheap, and it gives us immediate, actionable information that directly helps us save lives.
If you like this video, then please like, comment, and subscribe.